I'm Michael Brown. I'm Director of the Cancer Clinical Trials Unit at the Royal Adelaide Hospital. I'm a Senior Consultant Medical Oncologist. I'm most interested in cancer. That's what I do as a job. And I'm most interested in bringing the patient's own immune system to work against the cancer. That's the focus of my research. Three major things that have been the focus of research. First is the knowledge we're gaining about how to make the existing drugs on the pharmaceutical benefit scheme more effective and safe for cancer patients who receive them. The second is to take T cells from the patient's own blood, train them up in the lab, and then return them to the patient's body so that they can fight the cancer. And the third thing is to target radiation to the cancer so that the surrounding live cells can be killed, and that means then the immune system can see it better. And we've just started a clinical trial on the latter two things. The CAR T-cell therapy, we've had a trial going already. This has been in metastatic melanoma patients. We've treated 6. We went back to get approval to treat 2 more patients using a better product that we'd created in the lab. And we've now treated 2 of those patients. Because it's a safety trial, we can't really comment on how well it's worked, but it's continues to be safe. And we're also developing this same treatment for brain tumours, so aggressive primary brain tumours, glioblastoma multiforme in adults. And then there's a children's cancer called diffuse intrinsic pontine glioma, which is done as a collaboration with the Sydney Children's Hospital. So we'll be making the products for those children and for those adults, but those clinical trials are yet to start. The second major project is the molecularly targeted radiation, and that's apamab, and we have a phase 1 clinical trial already started. We've had 2 patients enrolled, and we're on our way to getting 18 enrolled altogether. So the T cells are taken from the patient's blood. Actually, just blood is taken. And there's a blood thinner put in. It's taken to the lab. They collect the white cells from that blood, and then they stimulate them in the dish. And then when they're stimulated, they're able to put into their DNA a virus that contains the training module, if you like, for the CAR T cells to recognise the cancer. So that gets integrated into the DNA of the cells. It's genetic engineering. It's a kind of genetic engineering. And that means those cells, when they're expanded further, they can then be given back to the patient to go and find the cancer and kill it. So if you take the apamab example, the next phase is actually to give this agent that will see dead cancer cells to patients getting standard chemoimmunotherapy for lung cancer. So this is now the standard first-line treatment for lung cancer patients. If it lights up, then we know we can deliver something with more therapeutic doses of radiation on it. That would be in a separate trial. And if we were able to do that, we could kill more cancer cells and get the immune system to see it better, and then kill even more cells. So we'd hope it'd be a virtuous circle of killing cancer cells. We've identified those patients in whom it's likely to work because they've already got dead cancer cells for the antibody with the radiation to bind to. So it's going to bind there and nowhere else. So that's why we believe it'll be safer and therefore more effective. We hope to bring technologies that are not currently in South Australia here. We hope to bring talented researchers here who can work with us on these technologies to apply them for the benefit of patients. We hope that this agency will be part of a network of national cancer centres. There's some idea the federal government might be favourably disposed to such a national network. I think we want to bring cancer care and the standard of it in Australia generally to a higher level as holds in other countries like the United States or the United Kingdom. So there has been an overseas analysis done for melanoma, so there is now some impact on the survival of patients with metastatic melanoma, but that's because some years have now evolved since the commencement of the treatment, really in the last 5 to 10 years in most centres, so you can start to see the outcomes. In this state, I don't think any analysis has been done, but I know I've got patients alive who wouldn't have been. So you would have to say it's had an impact. The other striking thing to say with melanoma, because it really is at the vanguard of all of these changes of using the immune system to fight cancer, is it's being brought back earlier into the disease, so that now many people will have had surgery and they in former times would have been fixed. That's it, we'll follow you up with scans. But now you can get some additional treatment and immunotherapy that can improve the chances of it not coming back and maybe even make you live longer. One of the drugs now on the PBS, there's data supporting that idea. And this is quite a different approach to treating cancer because chemotherapy had only formerly been used after surgery. In fact, it's being turned on its head where we're using the immune system before the patient even has the cancer removed to see if we can switch it on to kill off the cancer and then remove it. And if you do it in that sequence, you're less likely to have had it spread and grow back because those spread cells are killed off by the immune system at the same time. It's almost as if there needs to be cancer there for the immune system to see it better, kill it where it is in the lump, but also go elsewhere in the body, find it and kill it there too. Better if it's done before, it seems, than after, at least in mice.